Hello, this is Dr. Paul Cottrell, and I'm recording this on 5/7/2026 at approximately 11:22 a.m. I'm going to be going over a series of smaller videos going into detail about the hantavirus and what you need to worry about and what you don't need to worry about. And we're going to go into some of the biology, some of the mechanisms, and why I believe that there is a very low probability of a pandemic. But there has been research going on in the laboratories in Canada, in South America, and with Fort Detrick in preparing for the potentiality of a higher transmissible hantavirus that is human to human transmission. All right. So it's important before we get to that type of detail, we need to understand what exactly is a hantavirus and how it works. This video is dealing with the mechanism once it gets into the cell. So this is a cellular mechanism. So the hantavirus is a virus that it docks to the surface of the cell and attaches to a receptor. There's two types of attachment that we're going to go into later, but it'll attach to a receptor, and that will allow for the virus to come into the cell. So there's an endocytosis, and it creates an endosome, and that virus being attached to the receptors ends up starting to break apart. There's a change in the pH in the endosome, and that will lead to two different stages, an early endosome stage and a late endosome stage. The early endosome stage is still encapsulated. The hantavirus is encapsulated in the endosome. And then in the late stage, what happens is it starts to peel away and crack open the envelope of the virus and allow for the genome to come out. Now, what is interesting about this genome is that it is a negatively sensed RNA, single-stranded, and it has three strands. It has what is called the long strand, the medium strand, and the short strand. We'll go over the details of the genomics in a different video. Now, once the late endosome peels apart the envelope of the hantavirus, that actual genome, those three small genomes, start to move out of the endosome and into the cytoplasm. Now, what can happen? There are a few things that can happen here. Now, I'm going to jump forward and then jump back. There are two ways that the cell can exocytose the hantavirus. There's the packaging of the hantavirus at the surface of the cell, and then there's the packaging of the hantavirus through the Golgi apparatus. It has been shown, and I will do that in a different video, that mutations allowing for replication and packaging to the cell surface allows for more transmissibility for humans relative to the Golgi apparatus pathway. All right. So with that said, let's go back. So we have a late endosome that's peeling away the surface or the shell or the envelope, whatever term you want to use, to break up the hantavirus. And then the genome is released into the cytoplasm. And what happens is that, remember, there are three single-stranded negatively sensed RNA. That means that they have to go through a process to be able to produce proteins. So this is a negative viral RNA, so negatively sensed. All right. And there are N proteins that are protecting the genome. They start to break away. They're disassembled. The N proteins are disassembled. So now you have you have this negatively sensed RNA, and that can go through two processes. It can go through a replication phase, or it can go into a phase where it's going to turn into a messenger RNA, so a positively sensed. Now, let's talk about the replication phase first. So you have the negatively sensed single-stranded RNA of the long, the medium, and short genomes. And that will go through a replication and multiply, and they're all negative, and that multiplication is going to be negatively sensed. And that negatively sensed is going to be transferred or chaperoned to the cell surface and package with the glycoproteins. And there's two types of glycoproteins, the GN and the GC. And we'll go into details later in another video. Now, the other pathway is to take the negatively sensed RNA and for it to be turned into messenger RNA, positively sensed. So there is a transcription, and what happens is that it will dock to the endoplasmic reticulum. That messenger RNA will dock to the endoplasmic reticulum, and you have ribosomes on the surface of the rough endoplasmic reticulum that will actually start code for proteins. And now it's coding for the glycoproteins GN and GC, and helping with the cell, the membrane. So it's going to be coding for all the proteins that the genome is designed to produce. And then once those proteins are created, they are chaperoned to the Golgi apparatus and then assembled. And once that goes through a series of processes, then you will have a hantavirus that is going to be exocytosed. It moves to the surface and exocytosed. So the key here is there's two different versions of exocytosing. There is there's the process of packaging or assembling the hantavirus and the viron through the Golgi apparatus or through the packaging at the surface of the cell. Now, it has been shown that the packaging to the surface of the cell seems to be more transmissible for human-to-human transmission. Okay? Now, that covers the cell cycle as the hantavirus goes into the cell, how it gets processed, and then how it exocytoses out. Now, in terms of this negatively sensed multi-strand genome, when you have situations like this, when you have a long, a medium, and a short, and you have a different hantavirus coming into the same cell with some mutations in one of the genome strands, you can have something called genetic shift. So when you're talking about mutations, you can have genetic drift, where there's just some sort of amino acid changes in the code for the RNA. So the RNA is mutated in such a way that it's translating for some new shape for the protein. So that is what is called a genomic drift. But you can have replacement of these and swap of, let's say, the medium genome to the median genome of another hantavirus that is in the same cell or in the same tissue. All right. So if you have lots of cells in a tissue and you are infected by two different hantaviruses, it is possible to have a swapping of these genomes. Very similar to the swapping and the genetic shift in influenza viruses. Okay. So this is an added feature that is not present within SARS-CoV-2. SARS-CoV-2 is a positively sensed RNA that is one genome, not three genomes. All right. So that will give you a better idea that mutation and shift in the laboratory could be more prevalent. You can force cells to be infected with one type of hantavirus, and another hantavirus that has some type of mutation within, let's say, the medium strand for the genome could be infecting that same cell. And through just randomness, the packaging, either at the Golgi apparatus or the packaging within the cytoplasm, by up towards the surface of the cell, you can have a swapping of genomes and get the genetic shift. So it is actually easier to actually create different variants of the hantavirus in the laboratory. Now, in the wild, in nature, if your cells are infected or an animal cells are infected with different types of hantavirus, you will eventually get this swapping and genetic shift, just like influenza virus, because it has two genomes for influenza. This has three genomes, a large, a medium, and a small. Right? That's the key takeaway here. So again, it's low probability of a pandemic, but there has been research going on in the laboratory modifying the genome that is playing around with the GC and the GN. And I think it's GC and GN. Well, I'll have to, I think it is. Hold on. GN and GC, right? And then, and modifying the spike of the GN and the GC, or modifying the genome in such a way where the chaperoning pathway is more prevalent for assembly at the cell surface versus the assembly through the Golgi apparatus. So that's the key takeaway here. Thank you for listening. Please help support the work that I do. All these videos are for free. Please be a paid member to my Patreon channel. If you'd like to help support my work by donating, you can donate on my website, the-studio-reykjavik.com. You can donate through Stripe, PayPal, or buy me a coffee. And please take a look at the great products that I offer and help support the work that I do. Share the links. I'm going to do another video about some of the more of the details of hantavirus. Thank you for listening and have a nice day.